My name is Stephanie Seneff, and I have interviewed Eleanor in a delightful podcast interview where we talked about a lot of things, mostly focused on glyphosate, which is my passion, but also a lot about deuterium. And deuterium and glyphosate are synergistically toxic in ways that we describe in this episode. And I think you will learn a lot. We also talk about ways you can help to protect yourself from glyphosate.
Welcome to the Dr. Eleanor Stein podcast. In this show, you will hear expert medical professionals, researchers, and people with lived experience share science-based, low to no cost strategies to help people with complex chronic diseases. Listen in for another dose of education, empowerment, and hope. Hi, everybody. I am Dr. Ellie Stein, welcoming you to the Dr. Eleanor Stein podcast. On this episode, it is my pleasure to speak with Dr. Stephanie Seneff. I'm sure she is known to many of you. She is a senior research scientist at MIT's Computer Science and Artificial Intelligence Lab in Cambridge, Massachusetts. She has a bachelor's degree from MIT in biology, a master's and PhD also from MIT in electrical engineering and computer science. She's published over 200 peer-reviewed papers and has spoken widely around the world. Most recently, since about 2008, she shifted the focus of her attention to glyphosate, and that's what we're going to talk about today, the pervasively used herbicide. And since she shifted her focus, she's published over 4 dozen peer-reviewed presentations on that topic. And she's published a book called Toxic Legacy: How the Weed Killer Glyphosate is Destroying Our Health and Our Environment. Dr. Seneff, thank you so much for doing this and welcome to the podcast.
Thank you so much for having me.
I'm going to ask you, so just for some of the listeners, I just want to say this is the second in kind of a 2-part series. You were kind enough last week, meaning the week before the recording date, to do a webinar, which we called Glyphosate 101. So it kind of laid some of the groundwork for people who don't know too much about glyphosate. And that can be accessed through my webpage under the webinars tab. So I'm gonna ask you a few of the same questions just because not everyone will be familiar with that information. And then I'm hoping today we can dig deeper into the science.
Sounds good.
So given that you've got like a computer science background, how did you end up researching glyphosate?
That is quite a jump, isn't it? And I have to say, most of my career, I wrote a lot of computer code to teach computers how to communicate with humans using natural language. We were pioneers in that space. And so really at 2007, I kind of jumped ship and I was concerned about autism. I kept seeing the rates go up every year. We're just diagnosing it more, it has to be. And they're looking at all this genetic stuff, which, genetics doesn't go up every year like that. I knew it had to be something in the environment. And I just felt like I could do some work just looking at correlation statistics. That's really where I started. And I didn't know what it was, but I looked at a lot of different chemicals that I was aware kids are exposed to and looking for correlations. So over time, you can see the rate of autism is going up. So you're looking for something that's going up over time. And that eliminates a lot of chemicals right there, just that fact. And I didn't think of glyphosate at all. I spent 5 years looking at other things, like fluoride and lead and acetaminophen and all kinds of other. And of course, the vaccines were of concern to me because of the aluminum and the mercury in the vaccines. And I didn't even think to look for glyphosate because glyphosate's safe, right? It was like it couldn't be glyphosate. But it just so happened that I was at a conference where Professor Don Huber gave a 2-hour presentation on glyphosate. I walked into the room not knowing what it was, and I walked out of the room feeling like I had my answer. It was that dramatic. It was so amazing. I could still almost be there, listening to him talk. And because I'd worked for 5 years, I knew a lot about autism. I understood it was a problem with the gut. A lot of the kids have gut problems. I was thinking it might be something in the food, it might be something in the water. I was thinking along those lines, something they're eating, something they're consuming. And then the fact that the glyphosate is all over the food supply, I was not aware of that at that time. We don't hear about it. It's like it's so safe that we don't worry about it. The government doesn't measure it. It's like that safe, which is really, really scary. When something is believed to be that safe that they don't worry about it, and then it is all over the food supply and they don't care. I mean, that is a very serious problem. So I think the fact that it is so pervasive and that it is going up over time, exactly in step with the rise in autism. And then he showed all these different ways in which it disrupts the gut microbiome, it depletes the minerals, all these things that it was causing that I knew were going on in autism. I was just like blown away, to be honest with you. I just dropped everything else and started learning everything I could about glyphosate. Published my first paper on glyphosate like 6 months later, and which was well received and also, of course, controversial because I was saying this stuff is not safe, and it wasn't a message that anybody wanted to hear. So there was some pushback. Actually, they tried to get it retracted, and the journal stood tall and refused to retract it, which was great.
That's impressive.
Yeah.
Can I ask why it's considered to be safe? Is it that the research shows it's safe, or is it that glyphosate doesn't act on human cells, so it's just assumed that it's safe?
Well, it's some of both, but it's not quite correct because
Oh,
they
okay.
claim it doesn't act on human cells, whereas I think that's not true.
Okay.
And then, of course, our gut microbiome is very sensitive to it, and that's where the trouble lies. A lot of the trouble lies there because it disrupts the gut microbes, and that's how it affects our health. But they do make this big claim that it disrupts an enzyme in the shikimate pathway. We don't have that pathway in any of our cells. That's wonderful. This is the pitch that they make to say this stuff is really safe. And they did some tricks during the evaluation process, like they studied glyphosate in isolation, but they used it in a formulation that had a lot of stuff in there that's also toxic and also makes glyphosate more toxic. So they protected themselves during the study. Yeah, and then they look at the GMO Roundup Ready crops. They have all these crops that are resistant to glyphosate. They studied them organically. They didn't put any, and when they tried to see if the GMO was safe, they didn't put any glyphosate on them. I mean, that's just like so ridiculous because of course you're going to put glyphosate on a GMO Roundup Ready crop, right? Now they studied blades without glyphosate and said, oh yeah, they're fine. I mean, so they cheat all over the place in terms of how they do the studies. They also declared that you only had to look for 3 months at an animal study. And if you didn't see any obvious damage at 3 months, you were good to go. Like that was enough of a time period to evaluate for animals. Well, it turns out glyphosate's a slow kill. So Seralini repeated their study, exactly the study they did to get approval. One of the studies they did, he followed the same protocol, except that he kept going after 3 months. And he was giving them a low dose of glyphosate. These were rats, low dose of glyphosate for their entire lifespan.
And which is what we're getting. We're getting bits every single day. Yeah.
Yeah. And then they started seeing effects at 4 months. They did 3 months, it looked pretty good. They were still okay. 4 months, they started having problems. So by the end of their life, they had a shortened lifespan. The females had massive mammary tumors. The males had kidney problems and liver problems. There were reproductive issues. They found lots of problems after the whole life exposure.
So partially you're saying it's because the research was done in such a way to minimize what was found.
Absolutely. And this is what the industry does. They're very strategic in the way they design their experiments, to be able to get past the regulatory process.
Okay. So maybe this is a great segue to just dive in. So how does glyphosate affect us as humans? And like you've already hinted, part of it is through the microbiome.
Yes, I think quite a bit of it is through the microbiome, but not all of it. And this is where we can get into the issue of the glycine analog, because that is very crucial. I just want to say about the glycine analog right now, there was a study where they looked at 1,000 different chemicals that were tweaks on glyphosate, just do something a little bit different, but a very similar molecule. All of them failed. None of them worked, only glyphosate, which says that there is something very, very special about glyphosate. And none of those other molecules were glycine analogs. And that's the big red flag, glycine analog. That is a crucial aspect of its toxicity that they don't like to talk about, But glycine is an amino acid. It's the smallest amino acid. It's one of the coding amino acids, so it goes into all the proteins by the DNA code. Super important. It's also a neurotransmitter. And it also is it's a key factor in the methylation pathway. So there's lots of ways in which having trouble with glycine, disrupting glycine in the system is going to be a problem. And glyphosate does that.
And so I just, like my little simplistic brain, if it's actually replacing glycine, so that means every protein in our body has glyphosate in it.
This is what I say. This is my theory.
Yeah, guys, that's like the implications of that.
The implications are astronomical, and that's what I just want to show my book here, Tasha.
Yeah.
This it goes into a great deal of deep. That's the center core of this book.
Okay.
Is my argument that glyphosate is the glycine analog, and it is a glycine analog. There's no question.
Yeah.
The fact, the idea that it substitutes for glycine during protein synthesis is something they've pushed back hard on. So they're trying to, portray me as a conspiracy theorist, because it's not true. They're saying, even though their studies, and I have a chapter in my book where I talk about their own studies, where they've really convincingly showed that the only way you could explain what was going on is to say that it's substituting for glycine. There were very striking experiments that the industry has done. Even in fact, the GMO that's resistant to glyphosate, the way they make it resistant is they found a version of the protein that doesn't have a glycine at the active site. You switch out the glycine, you put in alanine, it's safe. It's completely safe. It's a complete switch, which is also very unusual because usually if you have something a little different, it'll be less, but it's not less. It's completely not. Like this version of the enzyme that's a microbial version that they put into the GMO crops doesn't have the glycine at the active site that the original EPSP synthase has, which is what makes it susceptible because I think the glyphosate is substituting.
And are there any studies actually showing that glyphosate is in our proteins or in the proteins of animals that have been fed glyphosate?
Well, a good study is one that was commissioned by Monsanto back in the 1980s. They did bluegill sunfish, and they fed them radio-labeled glyphosate so they could track the radio label independently of the glyphosate. And then they looked at their tissues to see if there was any radio label, and there was in the tissues. And then they tested for glyphosate using the normal glyphosate test. It came up short, like only 20% of the radio label could be accounted for as glyphosate. So then they got the brilliant idea of breaking down the proteins into individual amino acids and try again. So they added some chemicals that would break the proteins apart, and then they tried again. They got 80% yield. So they got a lot more of the glyphosate recovered, and they said, and perhaps it was incorporated into the protein. That's their words, not mine.
Okay.
So
But has there been research since then? Like, it seems like that's a pretty important study.
I know. And in fact, there was a paper that was published that has the title something like glyphosate does not substitute for glycine during protein synthesis. It's like this incredibly strong title. And when you read the paper, you find out it's got this figure with these boxes. Each box is a particular case where they found glyphosate substituting. And they gave a specific sequence, the specific amino acid sequence where they found a substitute for glycine of glyphosate because it was heavier. They actually did this. You can do this technology. I mean, it certainly can be done. I've got friends who are trying to repeat that experiment. So they claimed it could not, based on their study. They actually found it in maybe 12 different proteins, found sequences. I looked all those sequences up on the web because you can find out which protein it is. Many of them were in proteins that bind phosphate, which is the thing that I argue is a susceptibility feature of a protein, to bind phosphate. Many of these proteins bound phosphate. And then they had glycines in situations that I would predict would be especially convenient to put glyphosate in place because you have to have your neighbors have to be small because glyphosate has extra stuff that needs to fit. These particular sequences that they found matched very well with my concept of where it should be susceptible. And but then they found them equally in both the treated and the untreated group. And then they said, oh, well, maybe then it's just chance. Like it's all garbage. All of it's garbage is what they argued. But the fact is that both groups of cells, they were breast cancer cells. They'd been maintained in culture for decades. They came from some woman who had breast cancer many, many years.
HeLa cells.
Yeah,
If you read the biography of that story of how those cells got, it's kind of horrible, but
I know they've maintained
really without her will.
Exactly, I know. I remember that
Her
story.
consent. Yeah,
And they've probably been fed glyphosate in the nutrients all that while. So I think both groups had lots of glyphosate all over their proteins, and then they were metabolizing their proteins, making new proteins, and just grabbing that glyphosate and putting it back in. So I think, and you can even design an experiment to fail where you put glyphosate into the medium, but you design it such that the glyphosate doesn't get taken up. You can do that. And I don't know if they did. I'm not saying they did.
Yeah.
But it's quite possible that they were equally contaminated with glyphosate, and therefore both groups, had it. And they had another thing they were looking for. I forget exactly what it was. They thought they could also detect some other transformation, which they found none of in either cell type. So that one worked, like there was nothing. But with the glyphosate, equal representation from both sides. So they said all of it's garbage. And I think they made the wrong conclusion.
Okay. So one possible, I guess we would say not definitively proven mechanism is that it's substituting for glycine and therefore impairing protein and enzyme function. What are some of
If it does, then that makes sense as far as explaining all these diseases that are going up dramatically, because it's a huge list. And that's what I was doing at first, is just looking at correlations.
Yeah.
There's a huge list. And I can figure out which proteins. It's a big puzzle to solve. And I've got a whole list of proteins that have what I call a glyphosate susceptibility motif, binding phosphate at a place where glycine is highly conserved. And these proteins, when they're defective, are associated with these diseases that are going up dramatically in step with glyphosate. Like the whole thing fits together so beautifully that I think I have to be right.
Okay. What are some of the other mechanisms? And then we'll move into kind of what are some of the consequences. Yeah.
Even without that, you can still argue that glyphosate would be a big problem. And that's because of the gut microbes. And there you're in much much more solid footing, because there are many gut microbes that have this shikimate pathway that use it to produce crucial nutrients for the host. And they have the EPSP synthase, and many of them have that version of it that has the glycine instead of the alanine at the active site. They are susceptible to glyphosate, and that's been shown experimentally. And it's been, in fact, the first paper that I mentioned that I published with Anthony Samsel, I think in 2013, we talked about inhibiting two different groups of microbes, the lactobacillus and the bifidobacteria. And we argued that it would inhibit them. And then later papers have shown that, in fact, it does inhibit those two species in particular. So it's nice to see new papers being published that are validating what we said back then, in our paper. So we were more hypothetical, but it's now being proven that that's the case. And you would predict it, just given that they have that enzyme that has that susceptibility. And so it tends to kill off the beneficial bacteria.
I was just going to say it's not an equal opportunity employer.
That's a very good way to say it.
Okay.
I like that. And so the pathogens get a foothold, and then you get the inflammation, and you get the immune cells coming in, and then the inflammation really messes things up, because there's all these anaerobic bacteria that are really, really essential for good health. And when there's inflammation, they don't do well. And then so the anaerobic microbes get killed off. And then you've got all kinds of issues with insufficient butyrate. You've got gut dysbiosis, you've got inflammatory gut, you've got leaky gut. Inflammatory bowel disease is one of the conditions that's going up exactly in step with glyphosate. Also celiac disease, which is an autoimmune disease, with the gluten. Gluten is a very, very widespread glyphosate contamination in gluten-containing foods. And the glyphosate disrupts the bacteria, the lactobacillus, that have enzymes that help to break down the gluten. Gluten has a lot of proline, and proline is difficult to manage. It requires special enzymes that are produced by lactobacillus. So the lactose get killed off. The gluten doesn't get properly digested. You've got these short peptides that are allergenic. And then you get the immune cells responding to that with antibodies that then, through molecular mimicry, they attack human cells, human proteins, and you end up with celiac disease.
Oh, fascinating. And so basically it's taking out the good bacteria. Therefore, we're not getting all the benefits of those good bacteria. And one of the things you talk about is butyrate. Can you say a little bit more about, so butyrate is selectively made by the good guys. Is that
right? That's a good way to say it.
Okay.
You're good at having these analogies.
Thank you.
Yeah, the microbes that make the butyrate, they need a low pH. They need a low acid environment. And glyphosate creates a basic environment, raises the pH. And then they don't thrive when the pH is high, and then they don't make enough butyrate, and then the butyrate's deficient, and then the colonocytes get sick because that's their favorite food. they really depend upon butyrate as a source of energy. And when the butyrate is deficient, then the colonocytes don't do well. And then you get and there's also a problem with the mucin, sulfomucins. I believe the sulfomucins are badly disrupted by glyphosate. And I talk about that in my book. And that's partly because of the sulfation pathways, which are getting messed up.
And how does glyphosate interact with sulfur?
So the sulfation issue is mostly because of my idea about the substitution for glycine, because all the enzymes, many of the enzymes that are involved in managing sulfate, they're both synthesizing it, activating it, transferring it from one molecule to another, all the step of the way for the sulfation system. All of those enzymes have glyphosate susceptibility motifs. In other words, they have a perfect match for where you could expect glyphosate to mess them up. And that makes me think that glyphosate is killing that pathway. And that's also a great deal of what I talk about in my book. And of course, it makes sense also because sulfate deficiency is an issue in autism for sure. That's actually interesting. Both autistic humans and autistic mice, they've looked at them post-mortem and they found they have insufficient heparan sulfate in their brain ventricles. That's absolutely crucial because heparan sulfate is critical for the development of the brain. So when heparan sulfate is deficient, the neurons don't mature properly and the brain becomes disturbed. And that's for both mice and humans, that effect, And then also they have disrupted enzymes that make, it's the phenol sulfation pathway in the gut that gets disrupted. And that's a problem too for detoxifying toxic phenols. And so you have p-cresol, which is elevated in the blood in association with autism because it can't be sulfated. If it could get sulfated, it would become water soluble. It could be peed out.
Right. So I was just going to ask, why is sulfation in the body? So we're talking sulfur or sulfate?
Yeah, it's more sulfate than sulfur. But
okay.
A lot of people have sulfur sensitivity, and so they avoid sulfur-containing foods. Then they get a systemic sulfur deficiency problem. So sulfur is really important. It's underemphasized. It doesn't even have a minimum daily requirement. The government assumes there's going to be plenty in your food, but I think it's not true. And especially because glyphosate actually disrupts the supply of sulfur to the plants. So when you eat plants that have been exposed to glyphosate, they have less sulfur.
Oh, so it's a double whammy. I hate to use yet another.
A double whammy, that's right. You hit the plants and then it hits the host as well. So you get it from both the bacteria and the plants that you're eating that are insufficient sulfur supply. So that's also going on. And the sulfate, for example, acetaminophen. I think you probably heard about acetaminophen being linked to autism. I actually wrote a paper on that. I'm proud of myself. My first paper on autism, I talked about acetaminophen way back when. Even before I discovered glyphosate, I was aware of a possibility that acetaminophen could be a problem. And then glyphosate disrupts the sulfation pathways, and acetaminophen, especially in the infant, is detoxified through sulfation. So that's going to be also a double whammy, where the glyphosate makes the acetaminophen much more toxic than it would otherwise be if glyphosate is also present.
Right, because we use, for example, N-acetylcysteine to detoxify acetaminophen overdose, and I believe it's providing a sulfur.
That's right. That's right. Yes. But even sulfation itself. So both the glutathione and sulfate both can detox, but the sulfation pathway is stronger in the infant because the liver hasn't matured. So other pathways are not as available. And so they need the sulfation pathway more than older people do. And then glyphosate messes it up.
Okay. Are there also ways in which glyphosate, so I know you've reported that there are connections with cancer. So does that mean it's impacting DNA or causing mutations?
Yeah, the cancer connection with glyphosate is tricky and sort of more harder to argue. But I think it comes down to deuterium. And I really believe that glyphosate is a mess for the deuterium system, which is something I've become very fascinated with in the last few years. Since 2019, since December of 2019, I've been all over deuterium. And it's so, so fascinating. I'm loving it. And I'm really excited because I think there's a group of us that we call ourselves deuteronomics experts. And it's a very small group. We're quite tight, we do a lot of communication through email. And I feel like we're really onto something huge with the deuterium. And I'm very excited because you really can, when you frame metabolism through the eyes of deuterium, lots of things make sense that didn't before. And in fact, in terms of the contorted ways in which metabolism happens, because there's a lot of pathways in which something happens in the cytoplasm and then something has to get the substrate and the enzyme have to get taken into the inside of the mitochondria. And then an enzyme there does something to it. And then the product has to be taken out of the mitochondria. It's like,
Yeah.
why do they go through all this complication? Why can't they just do everything in the cytoplasm? It seems
Yeah.
easier, you know?
Yeah.
And when you look at that and you say, oh my God, of course it's deuterium. Almost every time that I find a situation like that, I can see that what's happening is that the inputs are going into the mitochondria and a reaction is taking place. And in many cases, that reaction produces deuterium depleted water. And that
So
is the
say
actual
more about
product.
again, for people who maybe aren't up on deuterium. I've been
I'm sure most people probably don't even know what it is.
rapidly upskilling on it in the last few months. But why is deuterium, what is it and why is it a problem? And then how does glyphosate impact that?
Great. Thank you for bringing me back to earth. So deuterium is actually heavy hydrogen. It's a hydrogen atom with an extra neutron. It's
Okay.
a natural element. It's everywhere in the world. It's like carbon-14 to hydrogen. You know, you have carbon-12, carbon-14, which has extra neutrons. Hydrogen has 1 proton and 1 electron, and deuterium has 1 proton, 1 electron, and 1 neutron, which makes it twice as heavy as hydrogen. And it's a natural isotope. It goes everywhere hydrogen goes, but it behaves differently, very differently because of this. It's twice as heavy, which is a big deal. With carbon, it's only 12 versus 14, so it's not so much heavier, you know. It's a milder effect. But the deuterium, wherever deuterium replaces hydrogen, things behave differently, and it really disrupts certain enzymes. There are certain enzymes that specialize in making sure that whatever reaction they're going to do, if that substrate has a deuterium sitting there that they need to take off, they won't do it. They just leave it aside. So they have this what's called a deuterium kinetic isotope effect. It's really fascinating. Some enzymes are remarkably good at noticing, oh my God, that's deuterium. I'm not going to touch it, you know? So then their product is low in deuterium because they make sure that everybody they pull in has hydrogen and not deuterium at that location.
And why would an enzyme be so picky? Like, why would the
Yes.
enzyme care?
That's an excellent question. And that's where the ATPase pumps come in because the
Okay.
mitochondria produce energy using this enzyme ATPase, and it converts ATP to ATP. Even people with basic biology knowledge know about that, right? These pumps produce ATP from ADP by adding an extra phosphate, and they make water at the same time. They take oxygen. This is the whole famous thing where you take glucose coming in and oxygen and carbon dioxide. Oxygen gets converted to water. Glucose gets converted to carbon dioxide. Essentially simplification there, but you end up with carbon dioxide and water when you metabolize various nutrients in the mitochondria. And that's in the citric acid cycle. They break them all down. But the last step is to convert ADP to ATP and to produce water from oxygen. And that water is extremely deuterium depleted. Because
And
they
we
make sure to
ignore
keep
that,
it
right? Like
Yeah.
we know about the ATP, which is our cellular energy, and we know that everyone knows you got to breathe oxygen, and we all know we're breathing off carbon dioxide. And before I started learning about deuterium, I totally forgot that water is produced.
Yeah, lots of water is produced in those mitochondria, and that water is deuterium depleted. And the reason why it is because everything that goes in to provide the protons that are going to make the water, everything's obsessed on keeping the deuterium out. And you see that so vividly once you're aware to look for it, and then you look at these enzymes, and there are all these enzymes that have that do proton tunneling as part of how they work. And whenever you've got proton tunneling, you're not going to get deuterium because deuterium can't fit through the tunnel. It's like too big, right? So any molecule that has proton tunneling, that's a molecule that's trying to get rid of deuterium. And there are lots of dehydrogenases that do that, especially in the mitochondria and also in the microbes. There's lots and lots of dehydrogenases. They're pulling hydrogen off of organic molecules, and they don't want deuterium. And they put it onto something called NAD in many cases. They make NADH, and that H is golden. It is a deuterium-depleted proton. And then there's an enzyme called NADH dehydrogenase, which you might imagine, it's also got this tunneling effect. It takes the H off of NADH and it gives it to the intermembrane space to pile up deuterium-depleted protons inside that membrane that surrounds the mitochondrion. And then those protons come out through those ATPase pumps. They go right through the pumps and they're driving. That's the motive force for the pump. And those are protons. If you put a deuteron in there, it's like putting sugar in the gas tank. It gets stuck, it can totally wreck the pump, but it'll definitely mess things up. And so as you get too much deuterium in those pumps, the pumps get sick. Mitochondrion gets killed, actually. It finally gets recognized as, oh my God, I've got too much deuterium. You need to get rid of me, and the cell knows how to clear out these mitochondria that have too much deuterium and start over, make a new one, They recycle the mitochondria all the time.
So how does glyphosate impact? So we've got this, what you're describing is this beautiful orchestrated dance that all of these cellular processes, in addition to their day job, are also removing deuterium from the places where it's going to be deleterious, let's say.
Exactly.
And are they putting it in places where we need it? Like just so there's a balance here, right? And my understanding is in connective tissue, we want deuterium because it helps make it stronger.
Especially in bone. And in fact, there's a really cool study on seals. Seals dive pretty deep in the water and they get a lot of pressure down there, so they need really strong bones. And they actually dope up their bones with twice as much deuterium as what's normally found. Really interesting. And we probably do that too, but probably not quite as well as they do it. But that's how you make your bones strong, is by doping them with deuterium. And the processes that do both of those things, I think, are disrupted by glyphosate. I mentioned the dehydrogenases, and they're super important in the microbes too. The microbes have a lot of dehydrogenases that are important for producing this deuterium-depleted proton supply. There was a study on E. coli that found a dozen different dehydrogenases that were suppressed by glyphosate in the study. There was a study on the soil bacteria, the bacteria that live along the roots of the plant. And they gave them a single dose of glyphosate at time point 0. And then they monitored them for 3 weeks. And they actually looked at, this was a wonderful paper. I was so happy to see this. They looked at the dehydrogenase expression collectively, all the dehydrogenases that these microbes produced over that 3-week period. And it kept on going down over the 3 weeks. It kept getting worse and worse because of that one exposure. So it looks to me like, and it makes sense because dehydrogenases bind phosphate at a site where they have at least 1 and sometimes 3 highly conserved glycine residues.
I was just going to ask, so those dehydrogenases are, which are just the fancy name for enzymes that remove hydrogen from molecules.
That's right.
We're constantly removing and putting them back, it seems.
Yeah, it removes them and puts them onto NADH in many cases, puts them onto NAD to make NADH. And then in the mitochondria, this dehydrogenase that takes it off of NADH gives it to the intermembrane space. So that's a whole system that is delivering low deuterium protons to the mitochondria to make the ATPase happy.
I got it.
So crucial.
And glyphosate interferes because these dehydrogenases have these, we'll say, vulnerable locations that glyphosate can substitute for glycine.
It exactly matches.
Not work.
It exactly matches my glyphosate susceptibility motif that I talk about in my book.
Got it.
And it's also been shown in human studies even, and in animal studies, that succinate dehydrogenase, malate dehydrogenase. And I think there's a third one. I can't remember which one that is.
Citrate
dehydrogenase. Yeah, I remember there's 3 of them that have been shown to be suppressed by glyphosate in animal studies. So it's not just the microbes, and it makes sense because they're called flavoproteins. And they have the tunneling effect, and they're going to get rid of the deuterium, and they're going to be affected by glyphosate because they have this whole business with that. The sites where they bind something called FAD, flavin adenine dinucleotide, and they also bind NAD, nicotinamide adenine dinucleotide. Both of those have lots of phosphates in them, and those sites where they bind have lots of glycine. So that it's actually a big setup because there's a motif that's a GXXGXXG or GXGXXG, 3 glycines, and then some wild cards in between. That motif is a characteristic of where proteins bind NAD and FAD. So you've got wonderful setup for glycines to be susceptible to glyphosate to mess up their ability to bind NAD or to bind FAD, which is going to ruin them. They can't tunnel. And then
Can you say something about tunneling? So, probably I'd say over the past year and a half, my brain is getting blown as I'm reading about, what's now being called quantum biology.
Yes.
And this tunneling is one aspect. So my understanding of quantum is, we've got all these biochemical processes going on. And now that people are able to measure them more precisely, we realize that can't be the whole story. There's certain chemical processes that we just can't explain. Like the energy doesn't add up kind of thing.
Yeah. No, it's very hard to understand, and I don't understand it completely. It's physics,
Yeah.
It's really tough. And they use this expression tunneling. There's an energy barrier that you need to get over in order to break a protein. You've got a proton that's bound very tightly to a carbon atom. They really love each other and they stick really hard, especially in these circumstances in these molecules. And so you've got to have a very clever enzyme to be able to pull that proton off. And there's an energy barrier that you need to get over before you can do it. It's like you have to get up over the hill in a way, is the way they describe it.
Right.
But apparently with the tunneling, you can actually just sort of tunnel right through the mountain. This is how they describe it metaphorically. It still doesn't make sense, as far as physics is concerned. It's hard to get your mind to understand what the hell's going on, just to be honest with you. But the tunneling effect requires that it be small. And so you can see if it's twice as big, it's just not going to tunnel very well at all. And so whenever you have a protein that takes advantage of tunneling, and it sets up a sort of very narrow channel. I mean, there's even something called a water wire, where you actually have these water molecules. And some of these enzymes have a very tiny space inside a hydrophobic region, and they let just one water molecule in. There's a hole that just barely fits a water molecule. If there's a deuteron, it's too big. You can't get in.
Right.
And they bring in these water, and they make a water wire. It's so cool. The water wire, if you imagine these water molecules are holding two footballs, right? And then, and then there's a football that's being passed down the chain. And so they get a third one. Now they've got three, and they need to get rid of one, So they ping pong the football down the chain.
Wow.
And the original one that comes in stays with the first one, right? He gets a football, he sends a football, gets a football, sends a football. The football that comes in never makes it through the chain. And especially if it's a deuteron, because they all have the heavy guy.
And the footballs are hydrogens.
The footballs are hydrogens. Yeah. So even if a deuteron comes in, they're like, I don't think so. I'm going to pass one of my protons and just hold on to this deuteron because it's too heavy and I don't want to bother to try to transfer it to the next guy. So they have these lovely water wires that keep deuterium out, because by the time you get to the end of the chain, somebody's held on to the deuteron.
Right.
But the end is where you care, because that's where you're going to put it on the NAD to make the NADH. And it's golden. You've guaranteed practically it's not going to be a deuteron. It's really
just for clarity, that once it's on the NADH, that's what kind of feeds into the mitochondria to make the energy.
So that's right. Then the NADH is going to deliver it to the intermembrane space through that NADH dehydrogenase, which is also probably suppressed by glyphosate, So the whole chain is affected by glyphosate. And the dehydrogenases are so important for keeping deuterium out of the mitochondria. And once you've got the deuterium there, you wreck the mitochondrion, you get all this reactive oxygen, and that causes cancer. I mean, so that is how I think glyphosate causes cancer, by disrupting deuterium. And in fact, deuterium-depleted water is a treatment for cancer that's had quite good success.
So I had Dr. Somliai on the podcast. His work is just brilliant. And again, there's this potential, really, I won't call it a solution in and of itself, because he really emphasizes, you use deuterium depletion along with all your regular cancer fighting treatments, but it's like it's not getting recognition, and his research is really impressive.
Yeah, I read his second book, and it's quite dry. It just talks about all these different patients, but the results are astonishing. And I really would think that people would notice this, and really want to get involved with this as a treatment, because it's not toxic. They take all these horrible drugs that kill their immune system, to try to kill the cancer. It's the wrong approach, by the way. I actually think cancer, and I have a paper on that. I think I have 2 papers on that I've done with colleagues, where we've argued that cancer itself, the tumor, actually hoards deuterium. And there's a lot of evidence. When you look at its metabolic policy, it's so different from normal cells. It uses glycolysis to get energy, and it uses the mitochondria to generate nutrients that can make DNA so that it can multiply, right? The cell needs to be able to clone itself. So it needs all that DNA to do that. And the mitochondria are helping it to make the DNA, but they're not making energy. That shuts
Right.
it
You
down.
can't do both at the same time.
Exactly. Yeah. It repurposes the mitochondria to do something else. And then it takes in lots of glucose and turns it into energy in glycolysis, which is an anaerobic pathway. It only gets a little bit of energy and passes out lactate. Lactate is actually a useful fuel for the rest of the body. So and lactate is a low deuterium nutrient. So the cancer cell is producing low deuterium nutrients and handing it over to the rest of the body. And it's hoarding deuterium. It doesn't mind if its mitochondria have deuterium because it's not making ATP. The cancer cells actually hoard deuterium.
So they're doing something protective.
Yeah, and they're delivering, and they even pump out deuterium-depleted protons using ATPase. There's another, the ATPase, that actually does the reverse of the ATPase that's in the mitochondria. It actually takes advantage of a proton gradient to push protons out the cell, not against the gradient. So in the case of the DNA of the ATP inside the mitochondria, it has to work against nature because the protons are more concentrated, trying to get them from a more concentrated region to a less concentrated. But in the case of the cancer cells are pumping protons out of the cell, and that's with the gradient, so it's easier to do.
Oh, okay.
But the ATPase pumps that do that, it's a reverse ATPase, right? It does the opposite because they pump protons across a membrane, but they're actually working in the situation where it's easier to pump them out because there are fewer proteins. I don't know if that's making sense to you. There are fewer proteins on the side that you're trying to get rid of, and so it's easier for them to go out. And those protons, and those pumps are able to they refuse to pump deuterium. So they're pushing protons out of the cell and providing them to the microbes, to the immune cells the immune cells swarm into the cancer environment, not to kill the cancer, but to take advantage of all the nutrients that the cancer is supplying, the lactate and the deuterium-depleted water. They're very happy to hang out there and get well. So I think the cancer cell is trying to fix the immune cells so they can work again, so that then once they're fixed, then they can kill the cancer. But they can't at first because they're sick. Does that make sense? I said that a little badly.
Yeah, well, that's definitely a different take. I guess just again to what I heard you say is like, we think of cancer as all bad.
Yes.
But at least in some regards, it's helpful because it's exporting proteum or normal hydrogen so that the immune cells can use it and flourish.
Yeah, and fix their mitochondria so that they are then prepared to fight the cancer. So the cancer cell is sort of the tumor environment is trying to set up a microenvironment for the immune cells that's a happy environment to help them heal. And then once
so what goes wrong then? Because we all know cancer tends to
No, I know. Well, what goes wrong, of course, is that if your liver is supposed to be doing a lot of things and it's got this massive tumor, they're taking over the liver so that the other guys can't really do their job, right?
Okay.
I really kind of thought about like a breast cancer. It could be a massive, like these mice that Glyphosate made these massive tumors. These mice had these massive tumors. They weren't necessarily cancerous, but they were tumors. And they were probably helping to solve the deuterium problem for the mice, and they weren't killing the mice. They were cumbersome to carry around because they were so big, but they weren't killing the mice, which I found interesting. So I think a breast cancer post-menopause, in a way, the breast doesn't have to do anything at that point, right? Its job is over. So it's a perfect opportunity to have a cancer that can help to solve the deuterium problem systemically while maybe not doing so much damage. You know what I mean?
So if we lower deuterium, like Dr. Somliai kind of advocates, even in healthy people for periods of time, to go on a deuterium-depleted diet to kind of hold the cancer back, then it wouldn't be needed. If we didn't have too much deuterium, the tumors wouldn't be needed to try to fix it.
Right. I think the deuterium
Wow.
is the reason why the cancer occurs, is because of the cancer cells are able to detect that there's too much deuterium, for example, in the blood. And then these cells become cancerous as a consequence of noticing that there's a problem. They're trying to actually rescue the rest of the body from what could be even worse than cancer. There are other things that you can get that are really bad. So like a blood clot to your brain, and causing a stroke. You don't want that, right?
Right.
So
It's so interesting. Like, we tend in medicine to be so like tunnel visioned, and we see things just in a reductionistic way. And when someone like you comes along and you just turn it on your head and say, well, actually, cancer might not be the problem, it might be the solution, although it goes awry.
yeah, it's a solution
It's
that's
like,
suboptimal.
yeah,
You don't want that solution. You don't want to have the deuterium problem in the first place. And that's where you need to get rid of the glyphosate, because the glyphosate, I think that may be its primary um, bad effect, is the effect it has on deuterium. I've reached
okay.
that conclusion lately, that might be the worst thing that it does. Now, it definitely messes up the gut microbes, and that definitely is a big problem.
Pretty bad too.
But that is part of the deuterium story, because the gut
Right.
microbes make very low deuterium proton. They make hydrogen gas, molecular hydrogen,
Right.
that's 80% reduced in deuterium. And then they use that molecular hydrogen as a reducing agent to make the butyrate. So the butyrate is low in deuterium. And so all those low deuterium nutrients get disrupted by the glyphosate because it's messing up those microbes. And then you start right there, and now you've got a problem that's going to grow.
So if we just go back a minute to the correlational charts, because I think I mentioned to you when we
Yeah.
talked before, like when I heard you talk years ago, I have to admit, I'll say it to your face, I was dismissive because I thought it's just correlations.
Yes.
And how do we know it's glyphosate? Because so many other things are also kind of escalating logarithmically. Like, we're getting less exercise, we're eating poor quality food. I know glyphosate is
Vaccines, I
that.
have to say.
Yeah, vaccines. We're not going outside. We're not getting sunlight. We're not touching the earth. Like, there's just so many things that in our modern lifestyle are
And
going awry. How
cell
can we
phones.
isolate? Yeah, cell phones, EMFs.
Yeah.
So how can you be confident that glyphosate is really having an impact and isn't what I call an innocent bystander?
No, I think you make a good point. And obviously, I always have to say there are other things that are also toxic that we're exposed
Yeah.
to. We really live in a toxic soup, And I also worry, actually, there's been rumors that Bayer may decide to stop making glyphosate because they're having so much trouble with these non-Hodgkin's lymphoma lawsuits, and they might just decide to stop making it. And if they do, then the question is, what will they replace it with? And there are all these other herbicides that are considered to be much worse than glyphosate.
Oh my gosh.
So you figure, if we don't solve the problem in a big way, just go back to certified organic, we may be worse off if they stop if they take away glyphosate from the market, and then you start using glufosinate and 2,4-D and dicamba and all these other herbicides that are supposed to be much more toxic than glyphosate. Where will we be then? So
Wow.
it's really it needs to be a much bigger issue. I think the glyphosate, to me, the fact that it is considered safe is what's causing them to ignore its level of contamination. It's in our water supply. I collected a rain sample in Cambridge, and I sent it to Anthony Samsel. He found glyphosate in it, so it's in the rain. Organic food is going to have glyphosate too sometimes, because they can't keep the rain out. It might be in the manure. There was a paper that showed glyphosate in the manure was a problem for organic crops. It's really hard to avoid it. It's just like so pervasive. They use so much of it, and they don't bother to find out how much is actually that we're being exposed to. The reason why I
It's
think
really
it stands
shocking that
out.
there's no records.
Oh, I
They're
know.
not keeping track. Like, that's
Our government doesn't bother to test at all. The Canadian government actually did a good job with over 8,000 food samples from Canadian and imports from US, from Mexico, from Europe, and they found a consistent story. My friend, I've lost his name, Tony Mitra. Tony Mitra is a friend of mine, and he's a Canadian citizen, but he grew up in India, and he harassed the government for many years, and they finally decided to go ahead and test these 8,000 food samples. It gave him the data, and he published a book that he called Poison Foods of North America, because it's U.S. and Canada stood out against Mexico and Europe. They had much lower levels in general in their foods. So it's very disturbing that our government has no concern about the glyphosate levels, no concern whatsoever. And they think glyphosate is essential for our agriculture. They think we can't live without it. This is the way they see it.
Right.
Which is
That
really,
the whole economy, like we won't be able to produce enough food without
we'll
it.
starve. Yeah, we'll starve.
What do you think about
Will
that?
go through the roof. I don't think it's true. I think that people are smart. Humans are smart, and they're designing. Technology may actually be a crucial part of the savior, because they're building these fancy tractors now that are run on solar energy and have vision, and they can recognize. They can know, oh, that's a weed, and they can carry some boiling water, and kill the weed with boiling. Would be very selective to direct the. It seems to me like one could, if we really cared, we could come up with a way that wouldn't require hiring lots of people to pull the weeds, because that's the thing we don't want to do, right? We've already, the number of people in agriculture has gone way down. It used to be like half the population were farmers, and now it's only like 3 or 4%, I think. So
and most of them are foreign workers, like in both Canada and the U.S.
We're getting rid of
most people don't want. Most people don't want those jobs for whatever reason, and so in Canada, we're totally dependent on so-called temporary foreign workers.
Yeah, I know. It's going to be a problem here where we're cutting back on the immigrants. I think we're going to see a shortage of people who are willing to take those jobs, which is the opposite of what we need right now. So that's of concern.
Can you say more about kind of the scope of health problems? Like I've seen many of your beautiful correlational charts, but maybe just the scope of the problems that you think glyphosate is contributing to.
Yeah, well, some of them have been very well characterized, because, like, for example, fatty liver disease is one that's correlated. And there's 2 studies that I know of, one on mice, one on rats, I think, and one on humans. And the rat study had levels way below the regulatory limits, so very low dose glyphosate for the rat caused fatty liver disease experimentally. And then the humans, it was interesting because they had these humans that they binned into 3 groups. They had the ones that didn't have any fatty liver disease that you could find, mild cases, and then severe cases. So they had these 3 bins, and they were able to show statistically significantly different levels of glyphosate in the urine. I was really surprised by this. Statistically significant among all 3 groups. So even the mild versus severe, the severe had more glyphosate than the mild, statistically significant. And then, of course, the ones that didn't have the fatty liver had the lowest levels.
Yeah,
I think that's really striking. So that, to me, is not correlation; that's causation. And then, of course, obesity is another one. Every time I can see around the world, when a country starts adopting Western food, and the fast food, of course, the fast foods have lots of issues in terms of deficiencies, nutritional deficiencies, as well as, of course, toxic chemicals, not just glyphosate, right? There's lots of insecticides, and there's all kinds of stuff on the dyes, the food dyes,
Yeah, and all the chemicals and
Yeah, and even the hexane that's used to make the oils. I mean, there's lots of chemicals that go into the processing of processed foods. Anytime you have something that requires a lot of processing, it probably also has chemical contamination to go with it.
So what about autoimmune diseases? You've kind of actually talked about 2 of them already, inflammatory bowel disease and celiac disease, and also neurodegenerative diseases.
Right, absolutely. Well, Alzheimer's, Parkinson's, those are both have correlations, very strong correlations with glyphosate in terms of the increase over time. It's shocking, actually, what percentage of older people have dementia or Alzheimer's. It's just shocking right now, and it's going up. It's really a shame that the elderly are losing their minds.
How do you think glyphosate might be impacting? Like I know, for example, in Parkinson's, there's pretty strong evidence that environmental toxins like pesticides are involved. I don't usually hear glyphosate named in particular.
It is, though. In fact, there was a study on exposure to farmers where they looked at all these the farmers, of course, use lots of things, so you have a lot of confounding factors. And they looked at all these different chemicals that they were using and looked for correlations and found correlations with a number of them. sort of like 1.2, increased risk. If 20%, I guess that is 20% increased risk for a whole bunch of these chemicals. And then they actually did a correction process where they tried to correct for what else was there present whenever you had two things together, sort of take that into account and try to correct for that. And pretty much everything else went down and glyphosate went up when they did that. In other words, the glyphosate was getting more of the blame when it was combined with the other ones. So I think that glyphosate is probably the worst one with respect to Parkinson's. That would be my guess.
Oh, wow. Okay. So what can people do? You've painted it into a different planet. Yeah. So individuals, we've got the level of the government level where obviously we want governments to measure glyphosate, do this, fund the studies that are needed to prove definitively, is it really a contributor or not? Like we need that research desperately. But what about, that might take 20 years. We know how that goes. So what about individuals that are listening to this and hopefully they're still listening? They haven't gotten totally overwhelmed and depressed. What can people do?
Yeah, obviously certified organic diet is very important. And my husband and I have been buying only certified organic at the grocery store now since 2012. Ever since I discovered glyphosate, we switched. We did a big purge and threw away everything and started over. Once I realized the significance of glyphosate. And I feel like our health has been better because of it. So certified organic is great. It's good that you have that label. It's not perfect. And the certified organic foods do have glyphosate in them sometimes. Certainly the studies in Canada showed that. Generally much lower than in the non-certified organic. So it's definitely better. Not necessarily perfect. And that's because, of course, the rain, the manure, even if they're not cheating. And of course, people could be cheating because I don't know how careful they are to make sure how good the process is to make sure that it's certified.
Like the farmers might be cheating and using chemicals when things are they get stuck. Yeah.
Yeah. And they might just manage to get by with that because there isn't a lot of resource to make sure that they're following the rules. So it's a amount of trust involved. But still, certified organic, I think, is a really good thing to do. Of course, I also advocate a high-fat diet because fats are low in deuterium.
Say more about that. Maybe, say people aren't eating organic, so just regular conventional food. Fat is the lowest. And then what about the other types of food?
So it's kind of like the fats are the lowest, the proteins are in the middle, and the carbs are the highest. So in general, high protein, high fat, low carb will get you lower deuterium food.
Naturally, without taking any deuterium depleted water or expensive interventions.
Yeah, and I do take deuterium depleted water right here.
There you are.
I have a little bit every day, just a third of a cup, which I mix with 2/3 of a cup of water, regular water, to make something like glacier water. So glacier water actually is low in deuterium. And that's because the deuterium tends to stay with the ice. Deuterium always tries to stay with a more dense state. And you had talked about if you had distilled water, it might be that distilled water is low in deuterium because the water vapor would have less deuterium because being heavier, it tends to stay in the liquid phase. And that's how, of course, you've got those gases. The hydrogen gas that the microbes make is 80% depleted. And that's partly because the enzymes know how to keep the deuterium out.
Right.
But a certain portion of that is simply that the deuterium likes to stay in the more dense state phase.
So why do you drink deuterium depleted water?
It's just kind of that I feel like it must be better than not because it considering how important the mitochondria are.
Yeah.
And just considering too that it works so well for cancer and that it's non-toxic, it's sort of like, why not? It is expensive, and that would be a deterrent.
Very expensive.
Yeah.
Especially to fly it to where you live.
I know. And then, of course, plastic bottles, which they had bottles. But
no, you can buy glass, actually.
Well, the company that I buy from had been selling it in glass bottles. Of course, then you've got a lot more weight. Everything is
so it stopped.
Yeah, so now they only offer plastic bottles. It's too bad that it's so hard to ship water.
So you're not using it to kind of impact the effects of glyphosate. You're doing it as like a general health measure.
Yeah, and I do think because of glyphosate, you need to be very concerned about your mitochondria and whether they're going to be healthy. So they kind of need all the help they can get. So I figure,
Got it.
I don't know if a third of a cup is so little that it can hardly matter, but I just do it. I'm
And
so passionate about deuterium. I feel like I should, and I eat a high fat diet, and also a lot.
yeah,
Sulfur containing foods. I like cruciferous vegetables, onions and garlic. I
okay.
eat tons of onions and garlic. Almost
Yeah.
every food I make has onions and garlic in it.
And cruciferous means what?
Broccoli, cauliflower, cabbage.
I think kale is even.
Yeah, kale has a lot of sulfur. I think it's not
Okay.
cruciferous, but it's a good. It's a very nutritious vegetable. And then a salad every day. Vinegar is good, possibly to break down glyphosate. There's some microbes. Acetobacter is one of the species that some of the members of the Acetobacter family can break down glyphosate. So it's possible that
Okay.
sauerkraut or vinegar might. I say that weakly because it
Yeah.
hasn't been proven. I don't have any studies to say that, but it just seems logical to me that that's a possibility.
Do
Very
you
few white microbes can break it down.
Do you recommend that people get their glyphosate levels measured and or their deuterium levels measured?
Right. You can do both, and I have done
Yeah.
both. And I have to say, I had glyphosate in my urine despite organic diet for many years, so that's discouraging. It
Yeah.
was low level, but it was there. It was detectable, and I was disappointed because I try so hard.
Yeah. I'm the same. I've got, and mine wasn't even that low. I'm more in the middle, like 50th percentile. I'm like, wow.
Yeah, it's terrible. It's really sad that we can't really avoid it.
And are there
society.
ways of detoxing glyphosate? Like, are there any protocols out there or supplements? And I know that's a whole other minefield, but for most other things, like if you have heavy metals, people recommend chelation and
Right.
or yeah.
No, that's true. Naturopaths have been recommending fulvic acid and humic acid, which is these complex organic molecules from the soil. The claim is that glyphosate, they bind to glyphosate and then they can take it out through the feces.
I've always wondered, what are those molecules supposed to do?
Yeah, and they
Okay.
also might harbor enzymes that are sophisticated enzymes that could break it down. So what I think is great is if you've got microbes growing in your gut that can break down glyphosate.
Right.
Unfortunately, some of the ones that can break it down are toxic, are bad pathogens, like Pseudomonas aeruginosa.
Oh,
That
don't want that one.
that one's a really nasty microbe, and it can break down glyphosate. And I actually think that it may be a reason why it's become a problem in the hospitals, because it becomes a feature. It's just like the cancer, right? It
Right.
can do something really good for you, which is to get rid of the glyphosate. But at the same time, it does something really bad, which is to produce these metabolites that make you sick. So it's a compromised situation. And that's the same thing within the waterways. There's these cyanobacteria that can break down glyphosate. And when they do that, they release glyphosate has a nitrogen and a phosphorus atom. So those are both good nutrients if you can get at them. So once you break it down, then the cyanobacteria provide nitrogen and phosphorus to the algae, and in the algae, you get the algae overgrowth, and then you get a toxic situation with the algae.
Got it.
So
So it sounds like the humic and fulvic, I hope I said that right, substances, and that comes from peat, right?
yes,
Yeah.
it's basically organic matter from soil, probably aged soil, ancient soils.
And is there actual evidence, or that's more of a speculation that it might help?
I think so what's his name? Oh, shoot. I can't get the name, sorry. But yeah, there's a company that markets it and that
Yeah.
claims that they have done studies that show that it's beneficial for glyphosate. And then a lot of naturopaths recommend it.
Okay.
And it was also used in a cow study. There's a study on cows in Germany, I think, where the cows were sick and they had high levels of glyphosate in their urine. And so they gave them fulvic acid, humic acid, and sauerkraut juice. And then they showed that glyphosate levels went down, and the cows also improved their health. So that's kind of cool.
Okay, so there's some we'll call it preliminary evidence.
Yeah. If it's good for a cow, it might be good for a human.
And I had a question. We're just getting toward the end. When you take your deuterium depleted water, so I've been always unsure how to take it.
Mm.
whether you should take a little bit every day. When I talked to Dr. Somliai, again, the cancer researcher, he recommended depleting all water, like not just once a day, but actually all the water you drink.
And
Or
what
a
level?
3-month period. I believe to aim to make the water about 125 parts per
Okay.
million to bring down the body level of deuterium to below 130, around like 130 to 135 is what he thinks is ideal to prevent and perhaps treat cancer.
Interesting. Yeah.
And that's a lot of work, like to keep track of your water for the day and make sure you have, in a sitting
Well,
there.
I'm somewhat lazy, so I find it I'd like to have usually it's in the afternoon.
Yeah.
I'll get a glass of water and go out outside and work on my computer. It's a kind of routine thing that I have. And if I have a podcast interview, I have to do it earlier. So today, it's because it's convenient to have water for the interview, and I just make it deuterium depleted water. So, but I just do a third of a cup, 2/3 cup of regular water. It's easy, and it lasts a long time. I mean, it's super expensive compared to water.
Yeah.
But if you think of it as wine, it's not so bad.
That's a good point.
And of course, medicines are extremely expensive, like the GLP-1 analogs, You're ridiculous, like $1,000 a month or something.
Yeah. So, this was really packed with information. I'm wondering if you can just leave kind of like a little bit of a summary of how people, like you've talked about the keto or the high fat diet. You've talked about the sulfur containing foods because sulfur is being depleted by glyphosate.
Fermented foods. I did say apple cider vinegar and sauerkraut. Sauerkraut for the because of the possibility of them breaking down the glyphosate, but also just fermented foods in general, because those foods actually have low deuterium. The microbes are able to make low deuterium products. So fermented foods, I think, are good from the standpoint of low deuterium, and also just because those are microbes and it can help to keep your gut healthy.
Okay.
So I like, and that's my key things: high sulfur foods, fermented foods, high fat diet.
Okay.
And I'm not necessarily a carnivore. I don't eat lots and lots of meat, but I do eat a lot of animal-based fats.
Okay.
And I eat meat. I'm not a vegetarian. I just don't overdo the protein, I guess is the way to say it. And I don't eat much in the way of empty carbs.
Yeah. And I guess it would be safe to say organic, and also perhaps. So would you say if you buy organic grains, because grains, we didn't actually touch on this, but I was kind of horrified to find out, even if you avoid GMO foods, you're not avoiding glyphosate.
That's something people
Maybe just end with that, because that's
right.
pretty important for people to know.
Yeah, wheat and oats and barley and garbanzo beans and chickpeas and lentils, really high levels. Those were the highest, really, collectively, they were the highest levels in the Canadian study, I would say. Higher even than the GMO crops. And all those are non-GMO. It's because they spray right before harvest, especially in Canada, by the way, because you're fighting a frost.
Right. We're always in a rush in Canada to get the crop in.
Right. And either it's going to die from the frost or you're going to put the glyphosate on it to hurry it up so that you can harvest it before the frost. So that becomes crucial. And really a disturbing situation if you're in that pickle, as a farmer.
Maybe to end on a positive note, is there a resource that people could go to online that says, is a bit of a guide as to which foods are the safest and which maybe to avoid, especially if they're not organic?
Yeah, I've seen the sort of dirty dozen, which is not really correct, and the clean 15 and the dirty dozen. Those particular foods that they highlight have nothing to do with glyphosate.
Oh, okay. So is there anything glyphosate specific?
There are certainly. I mean, Zen Honeycutt has done a lot of. Zen Honeycutt is the founder of Moms Across America. She's a good friend of mine. She's done a lot of studies of different categories of food to see how much glyphosate is in them. And I mentioned the wheat, all the grains and the chickpeas, garbanzo, the legumes and the grains are going to be high. You really don't want to eat those non-organic, and none of them are GMO. And then of course the corn, the soy, canola, those are GMO. Also the sugar beets, so those want to avoid those as well. So
So moms across America, would that be a resource? Do they have?
Yeah, she has a lot of, she's done a lot of studies. She did a study
Okay.
on fast foods. They were all contaminated with glyphosate. She did a study on school lunches. 95% were contaminated. It's like it's all over the food supply. So that doesn't really tell you. It just sort of says don't eat anything. But
Well, it'd be
processed
nice to put a
foods-
link, right? So we're
Yes,
feeding
lots
people
across
with all
America.
this kind of
It's really great.
fear-inducing information, but it's nice to have a resource so that you can actually take action.
Right. Yes. Moms Across
Okay.
America is a really great site. And then I want to say Sunlight. I always like to mention Sunlight.
Okay.
I'm a big believer in getting out in the sun, and sunlight is very therapeutic. And we are way too aggressive in terms of trying to protect ourselves from the sun by slathering on sunscreen and wearing sunglasses. We shouldn't be doing that.
And how will sunlight, I know, without going into like a whole other podcast, but like Reader's Digest version,
Yeah.
how will sunlight help people in the context of glyphosate?
Well, it's interesting actually because sunlight stimulates. It causes the sulfate. So you have the sulfated glycocalyx lining all the blood vessels.
Okay.
With all these sulfates, and then the sunlight actually triggers growth of that sulfated. It makes the gelled water bigger in response to the sunlight, and populates more sulfate, which is really good because having a lot of sulfate aligning the blood vessels keeps them healthy.
And
So-
helps things move, like helps our blood.
Yeah, well, the sulfate makes the gelled water. The gelled water pushes out the protons. The protons are not deuterons, so it's supplying the sulfate. The gelled water traps deuterium and pushes out protons that are deuterium depleted into the circulation.
Wow.
So it's a terrific system for lowering the amount of deuterium in the blood by trapping it in the gel.
Okay. We'll leave it there. So we'll add sunlight is like the cherry on top.
Yeah, sunlight is great because it's free and it's comfortable. Yeah, I often hang outside, not necessarily in the sun, in the shade, outdoors, and work on the computer or something. So, luckily, I live in Hawaii, where there's a lot of opportunity to be outdoors.
Sounds wonderful. Thank you so much. This was really jam-packed, and
It was.
I think people have a lot they can take away from this, and especially even if the first part was a little bit complex and a little bit scary, I would say focus on the latter part where you're giving such practical steps people can take.
Yeah, great. This has been wonderful. Thank you for having me.
You're welcome. Thanks for joining me on the Dr. Eleanor Stein podcast. I hope you are taking away new strategies and ideas to improve your health and a renewed sense of hope. Join me, Dr. Eleanor Stein, for our next episode. Can't wait? Check out my website for a wealth of free resources.